Today, we are going to talk about the components of complement and complement activation pathways. This is a topic in the core paper 10, which is immunology paper, unit 7. So, the complement system is an enzyme cascade system, which is a collection of blood and cell surface proteins that help the abilities of antibodies to clear pathogens from an organism. This system comprises of about 20 different proteins that include serum proteins or serosal proteins and cell membrane receptors. It is an important part of the innate immune system. It alerts the phagocytes of the innate immune system. The complement system comes into play only when it is activated. The components of the complement system, that is the various enzymes, the major components of which are from C1 to C9, C1 through C9. The other components are factor B, factor D, etc. All these components, they are produced in the liver and distributed throughout the body fluids. These various factors which we talked about, C1 to C9, factor D, factor B, all these are enzyme precursors, that is, they catalyze a series of enzyme reactions of cleavage and activation to produce products of the complement system. So what is a complement? Complement are set of serum proteins or serosal proteins that cooperate with both innate immunity as well as adaptive immunity. Now, as I told you, the complement system needs to be activated, and depending on that, the components of the complement involved and the triggers or the antigens, the complement system can be divided into three different pathways. That is the classical pathway, the lectin pathway, and the alternative pathway. The end result of all is a massive amplification of the response and activation of the cell-killing membrane attack complex. So at the end of all the three pathways, membrane attack complex is formed. This membrane attack complex forms holes in the bacterial cell wall, and it brings about lysis or degranulation or phagocytosis, depending on through which pathway it acts. So here I have included a YouTube video which was provided by McGraw-Hill Animation. This is a small video about 2.5 minutes where you can get an overview of the complement system. This is an animated video. Okay, so on this slide we can see on a lighter note that the complement system can be divided into three types: classical pathway, alternative pathway, and lectin pathway. The three pathways differ in their initiation, that is, the activators. As you can see, the classical pathway is activated by formation of antigen-antibody complex. The alternative pathway is activated by the recognition of bacterial endotoxin, and the lectin pathway is activated by mannose, which is present in the bacterial cell wall. Binding of lectin to mannose results in activation of this pathway. Now we have to see all the pathways in detail. Here I have included another slide, another YouTube video which is showing us one more animated overview of the whole complement system. This will clarify your idea about the complement system. So let us start from the classical pathway of complement activity. As already discussed, the classical pathway of complement activity is initiated or activated by the antigen antibody complex. It is never initiated by a free antibody, a native antibody. Always an antigen antibody complex has to be formed. Now these complexes may be soluble or they may be formed when the antibody binds to antigenic determinants. Where are these antigenic determinants present? They are present on the cell wall of the parasite or the bacteria or the fungi, whichever is the pathogen, the disease causing organism. So basically they may be called epitopes. The main antibodies that are involved in production of this reaction are IgG and IgM. Various subclasses of IgG are involved in this reaction. In the next slide, we will see the activators of classical pathway, the various antigens which can activate this pathway. They may be C-reactive proteins, staphylococcal protein A, or factors produced by apoptotic cells or necrotic cells, etc. Here you can see the overall complement cascade. As I have told you in the introduction, the complement system is a cascade of enzymes where enzymes, basically proteases, they keep on either activating or by uh, breaking down other enzymes they function, and ultimately they produce C3 convertase. This C3 convertase activates C5 convertase, and C5 convertase leads to the production of membrane attack complex. So let us see what are the steps in the classical pathway. The classical pathway involves these components: C1 component, C2 component, and C4 component. The C1 component is actually a complex. It is a 774 kilodalton protein complex. It consists of a recognition molecule which is C1q, and it also consists of a tetrameric protease complex, that is C1R2S2, because it is a tetrameric complex. It has serine protease domains tightly packed in the center of this complex. What does this complex do? C1R2S2 tetrameric complex. It binds to IgG or IgM containing immune complexes. So as soon as the pathogen, the disease causing organism, enters your body, either IgG or IgM recognize it, bind to it, they form immune complexes. These complexes are then recognizable by the tetrameric protease domain of C1 complex through C1q recognition molecule. Now the formation and activation of the C1 complex causes cleavage of C4 component. The C4 component is converted to a C4A and C4B component. It also causes cleavage of C2 component into C2A and C2B. So C1 complex, C1 esterase on activation causes the cleavage of two components, C4 into C4A and C4B, and C2 into C2A and C2B. The C2B and C4B component combine to form a C3 convertase, as you can see here. This C3 convertase then combines with other components such as C3b to form another complex which is called as the C5 convertase. The components of C5 convertase are C4b, C2b and C3b. Once the C5 convertase is formed, it converts the C5 component into C5a and C5b. The C5 component is larger in size. This combines C5b component combines with other factors, other components C6, C7, C8, C9 to form a membrane attack complex MAC. So the whole of classical pathway can be summarized as such. First, there is activation of C1, which is the initiation step. C1 causes cleavage of C4 and C2. The components of C4 and C2 combine to form a C3 convertase. Why is it called C3 convertase? Because it again cleaves C3 into C3a and C3b. The C3b combines with the C3 convertase to form C5 convertase. Why is it called C5 convertase? Because it again cleaves C5 component. The C5 convertase cleaves C5 component into C5a and C5b. The C5b component with C6, C7, C8 and C9 join together to form a membrane attack complex. All these things have been described here. Through this picture also you can see. So ultimately a membrane attack complex is formed, and you can see in this slide the three-dimensional view of membrane attack complex. This membrane attack complex forms holes or drills into the bacterial cell wall, the cell membrane. Through this, now the macrophages can act or the mast cells can act, and they can bring about the lysis of the disease causing organism. So let us see now the second pathway of complement activation, that is the alternative pathway. The trigger for this pathway is bacterial endotoxin. What is bacterial endotoxin? It is a lipopolysaccharide found in the outer cell wall of gram-negative bacteria. The other triggers for this pathway can be yeast cell wall or cobra venom factor, etc. Now all these three pathways, they converge at the formation of C5 convertase. From C5 convertase, they are all common pathways. So up to that, there is difference in the pathway. The triggering and the initial steps are different in these three pathways. So the alternative pathway can see involvement of various factors such as factor B, factor D, factor H and factor I. The factor B and D factor are important here. So in the first step, the factor C3, it is converted to factor C3b and factor B by hydrolysis. Then in the second step, the factor B is cleaved into a BA and BB factor by the involvement